Good evening. My name is Priya Hill, and I am presenting my section of our presentation on the neurological stem cells and therapeutic applications in reference to Alzheimer's disease. In this presentation, I was responsible for the biomarkers, mechanisms, and the significant sections, so that is what I'll be talking about in this video. So, what in the world are biomarkers? In neural stem cells, the term biomarker refers to the various proteins and transcription factors expressed on the surface or within a cell. They can be used to identify specific stem cell populations and indicate their functions. Now, some of the main biomarkers we focus on include those that identify stem cells and the type of stem cell they are. So we have SOX2, Nestin, CAC6, DAC1. There are also those biomarkers that identify cells that are proliferating, so K167 and PCNA. Then we have those markers that tell us what type of cell a stem cell has differentiated into. So TUJ1, GFAP, and O4. We also have biomarkers that regulate niches of neural stem cells, such as those involved in the Notch or WNT pathways. We also have those biomarkers that monitor the effects of genetic modifications. So they basically tell you if a stem cell is still a stem cell. And we're looking for those lineage markers, which is basically, it tells us if a cell has differentiated or committed to a cell line, lineage. There are also biomarkers that allow us to track the engraftment and integration of neural stem cells post-transplantation. Typically, if you have a neural stem cell transplant, which I'm sure we'll talk about later in the presentation, in order to track those cells, the transplanted cells can be tagged with GFP, reporter protein. GFP stands for green fluorescent protein, and that allows us to see the original stem cells. GFP fluoresces and emits green. As the stem cell travels and mobilizes to wherever it needs to go, whatever niche it needs to go to post-transplantation, we can monitor the survival and stem cellness of that cell by looking for biomarkers such as K167. Identifying that K167 biomarker tells us that the stem cell is still actively dividing in whatever niche it has traveled to. So it's still being a stem cell. It still has all of those stem cell characteristics. In order to make sure that the transplant was successful, you can then look for the loss of stemness and gain of lineage markers. Biomarkers can also be used to evaluate the response to therapeutic interventions. Once again, we're looking for that loss of stemness / commitment to cell lineage or to a cell lineage. We've successfully established biomarkers and some of the uses that they can have. Now, biomarkers are plenty interesting. I'm sure you're just fascinated and would love to know more. Let's pivot to mechanisms. The WNT pathway promotes neurogenesis, which is responsible for the proliferation and differentiation of neural stem cells. This pathway works by having the external WNT ligand bind to a receptor, frizzled / LRP, and then that act of the ligand binding the receptor prevents the degradation of B-catenin, BCAT. BCAT then goes to the nucleus, where it activates a bunch of genes for proliferation and differentiation, and then these are the genes listed in parentheses. The next pathway is the Notch pathway. This pathway is responsible for the maintenance of stem cell dormancy and self-renewal. In this pathway, the Notch receptor on one cell binds a ligand on an adjacent cell, which triggers the Notch receptor to be cleaved. The enzyme that cleaves the receptor is called a gamma secretase. Once that cleavage occurs, the intracellular domain of the Notch receptor, called NICD, activates a bunch of genes that then inhibit neuronal differentiation. The next and final mechanism that we're going to talk about is the mTOR pathway. This pathway promotes growth and proliferation of neural stem cells. mTORC1 gets stimulated by various signals, such as insulin, and that causes the activation of protein synthesis, which is the cell growth signal. That cell growth signal inhibits autophagy. So why was all that important? Why did we sit here and talk about all those biomarkers and all these pathways and mechanisms? Why do we talk about that? In Alzheimer's disease, amyloid B plaques and tau tangles accumulate in the neurogenic niches of the brain. This accumulation causes degradation of pre-existing neurons, disruption of the normal signaling pathways, and the inhibition of neurogenesis. So that WNT pathway that we just talked about gets inactivated. Those amyloid B oligomers, they bind to that receptor, that original WNT receptor, and it can't bind. Okay, so that pathway, it can't bind to the ligand, so that pathway gets shut down. The Notch pathway, the Notch receptor doesn't get cleaved, so NICD can't activate the inhibition genes. That pool of NCSs decrease because they differentiate, because nothing's telling them not to. The mTOR pathway, that's hyperactive. So now toxic protein aggregates, amyloid B plaques, those are going to be allowed to accumulate. And then that constant cell growth signal is also preventing differentiation. So all of these pathways and all of these biomarkers and all of these things being affected and disrupted and blocked lead to the inhibition of neurogenesis in adults, which leads to an overall decrease in the number of available neural stem cells within the brain. If that number of available neural stem cells in the brain decreases, you're going to have less neural stem cells available for neural repair and replacement. So the damage is not getting fixed. The damage isn't getting fixed. That means the number of neurons, the number of healthy neurons you have in your brain is decreasing, specifically the ones in the hippocampus. If you're affecting the neurons in the hippocampus that are responsible for memory, the effect is going to be early memory loss. So that is the significance of these biomarkers and mechanisms in relation to Alzheimer's disease. And that is the end of my part of the presentation.